Now let's see how nano-drug delivery systems can only be deposited on the cancer tissues and how drugs are delivered to those areas. The characteristics that distinguish cancer tumors from normal tissues are used in passive targeting. As a result, no targeting ligands are used in passive targeting. On the other hand, active targeting makes use of targeting ligands that target specific cancer tissues. Passive targeting is currently used by the majority of clinically used nanodrug delivery systems. However, many active targeting nanodrug delivery systems are awaiting FDA approval. Now let's see first passive targeting. There is no targeting ligand on the drug delivery systems in passive targeting. The cancer cells are targeted by detecting physiological changes in them. First, we use enhanced permeation and the intention effect for passive targeting. This is called EPR effect. Angiogenesis begins when the size of the cancer cell exceeds 2 mm³. For rapid multiplication, cancer cells require large vascular roots to absorb nutrients. The cell is leaky and imperfect as a result of its rapid growth. There is also lack of lymphatic drainage. Because of the cancer cell's incompleteness, an external object can easily permeate the cell. The external object has a considerable retention time once it enters the cancer cell. As a result, nanodrug delivery system can easily penetrate a cancer cell and remain there for an extended period of time, continuing delivering drugs to the cell. The drug system should not be captured by the reticuloendothelial systems, body defense system, in order to deliver drugs to the cell. After the body defense system has captured the drug delivery system, it is removed from the body via the liver and the kidney. To avoid capture, the nanosystem should be less than 100 nanometer in diameter and coated with hydrophilic polymer. Hydrophilic coating prevents absorption on its surface, allowing the system to circulate in the bloodstream for a longer period of time. We can compare the diameter of tissue, cancer tissue, and normal tissues. As can be seen, the diameter of cancer tissue is much larger than that of normal tissues. Now let's see a tumor environment. The tumor environment can be used to permeate the nanodrug system into the cancer cell. The cancer cells have a unique affinity and have undergone physiochemical changes. We can utilize cancer cells' specific characteristics to get the nanodrug system into the cancer cell and deliver drugs to them. First, Cancer cells have a pH of less than 7.2, whereas normal tissues have a pH of 7.4. As a result, when the pH-sensitive nanodrug system is injected, it only interacts with cancer cells. The pH-sensitive polymer or liposome, the nanodrug system, is broken inside the acidic environment of the cancer cells, delivering drugs to embedded within it to the cancer cells. Second, the temperature of the cancer cell is slightly higher than the temperature of normal tissues. As a result, we can design a temperature-sensitive nanodrug system that exclusively interacts with cancer cells and delivers the drug to those cancer cells. As you can see here, tumor vessel is leaky, so nanodrug system can easily penetrate into the cancer tissues and remain there for a long time to deliver the drugs on its surface or inside it. So the same figure. So it shows the imperfect lymphatic drainage. Now let's see what kind of active targeting we use. Cancer cells may overexpress receptors, antigens, or proteins in some circumstances. If a ligand that matches the behavior of certain cancer cells is used, the nanodrug system can be observed by the cancer cell. The nanodrug system is observed by the cancer cell via receptor-mediated endocytosis. When the receptor on the cell membrane interacts with a ligand on the nanodrug system, the nanodrug delivery system penetrates cancer cells and delivers the drug to them. So this figure shows the penetration of the nanodrug system into the cell by receptor-mediated endocytosis. So if we use the targeting ligand, then if the receptor of the certain cancer cell is interact with the targeting ligand, then it can be observed by the cancer tissues, and inside it the drugs are relieved and the cancer cell can be eliminated. Now let's see what kind of targeting ligands are used. The first, folic acid. Folic acid is a vitamin that is required for normal cell function. The cancer cell requires a lot of folic acid since cell division is very active in cancer cells. On the cell membranes of lung, brain, breast, and ovarian cancers, folate receptors are overexpressed. Folic acid is thus used to target these cancer cells. Next, peptide. Peptide receptors of various types are expressed on the cell membrane of cancer cells. To target these peptide receptors, peptides are utilized. To target the peptide receptors expressed by diseased or cancer cells, peptides such as PGF, the vascular endothelial growth factor, a signal protein produced by cells that stimulate vascular genesis and angiogenesis. And VIP, vasoactive intestinal peptide, and LHRH. This is a luteinizing hormone, releasing hormone. This can be used to detect the cancer cells express the peptide receptors. Next, lectin. The cell type determines the type of polysaccharide expressed on the cell membrane. The well-known lectin hydrocarbon interaction is used to target specific cells. So we can use lectin for this. Antibody. In comparison to normal tissues, cancer cells over or underexpress certain proteins. Several overexpressed antigens on the cancer cell have been identified due to the advancement of proteomics. These antigen proteins are targeted by antibodies. Next, sugar. Galactose. This galactose is utilized to target the lectin that is expressed on epithelial tissue. The sialoglycoprotein receptor is found on the liver surface. This receptor is targeted with galactose. Next, lipid. The synthesized phospholipid interacts with the cancer cell membrane, causing it to change its lipid composition and permeability. The signal transduction in the cell is disrupted as a result of these alterations. The cell dies as a result of this dysfunction. To target and kill tumor cells, synthetic phospholipids such as edelphosine and methyl-phosine are used. This is edelphosine and methyl-phosine structure, chemical structure. These two lipids confusing the cell signal in their interaction with the cell membrane. These pseudophospholipids eliminate the tumor cells. So these lipids confuse the cell signal interaction and change the composition of the cells, and eventually this alteration cell is disrupted and finally eliminated. This table shows some other ligand, and these ligands targeting receptor and also targeting tissues. Antibody acception as targeting breast cancer cells. The receptor is antigen HR2 receptor. And this is the protein active in breast cancer. And the ligand folate. This folate is water-soluble vitamin B9, and targeting the folate receptor. And transferrin, this glycoprotein that regulates ion-ion transport, and targeting the transferrin receptor. This is used for the infected infected red blood cells. Galactose, which is called brain sugar, is targeting receptor of the asianoglycoprotein receptor. Thus, there is the liver cancer. Hormone EGF, targeting EGF receptor. This is epidermal growth factor. This control, this EGF receptor control the epidermal growth factor, and the hormone EGF targeting this receptor. Trypsin. Trypsin is immunological material synthesized in the spleen. And this trypsin is targeting the trypsin receptor. An anti-BFN antibody is targeting the neovascular tissue, and the receptor is VFN, fibronectin isoform. This RGD protein targeting newly formed capillaries, and that's related to the receptor of the alpha mu beta 3 integrin. Okay. The protein integrin is in charge of signal incomings and outgoings. Integrin is a receptor that is involved in the formation of new capillaries. Cancer cells have a tendency to form new capillaries in order to feed nutrients to the cells and allow them to multiply quickly. The angiogenesis inhibitor can stop the capillaries from forming. To deliver angiogenesis inhibitor to the cells, the RGB-based protein is employed to target the alpha mu beta 3 integrin. So this RGB protein is used to prevent the formation of the new capillaries. So there are various kinds of ligands, and these ligands targeting the receptors and target tissues.